Composition of soil fauna in the arena forest. Generally, soil fauna is comprised of bacteria, fungi, protozoans, nematodes, mites, collembolans, annelids, and macroarthropods. They are essential components of ecosystems, influencing decomposition and nutrient cycling. This list is by no means exhaustive, but serves to provide an idea of the immense biodiversity of the rhizosphere. Soil fauna composition is very understudied in Trinidad and Tobago, according to A. H. Strickland. They examined areas of vegetation in Trinidad and Tobago, including the arena forest, identifying soil conditions in several groups of soil arthropods. It was found that in every locality sampled, the Acarina formed the dominant group of soil and litter animals. Yang and Chen examined soil faunal composition of different forests across China and found very high soil faunal diversity. They found no variation in soil fauna richness and abundance between the three forests examined. Similarly, Wall et al. Maintains that in the tropics a great deal of attention has been given to above-ground organisms, while few studies deal with the diversity of invertebrates into soil, leaf litter, or dead wood. Further to this, Gonzalez and Barbena Arias point out that comparatively, data on soil biodiversity in tropical forests is very limited versus data on other ecosystems, such as temperate forests, grasslands, and deserts. Overall, there is a significant informational gap relating to soil fauna ecology in the tropics, which extends to the arena forest of Trinidad. To determine the soil faunal composition of the arena forest, collect selected and relevant environmental data using random sampling and quadrat sampling based on transects. Identify collected organisms and construct a species sample matrix using taxonomy keys. Use collected data and appropriate methods to determine absolute soil faunal density. These methods include extraction, preservation, storage, and labeling of organisms collected. Calculate ecological indices such as diversity, dominance, and density. Methods. Two 30-meter transect lines labeled 1 and 2 were set up. The general environmental conditions were recorded, such as cloud cover, cloudy, overcast, raining, sunny, relative humidity, air temperature, and wind speed. To achieve Objective 1, quadrat sampling for microfauna and macrofauna was conducted. Samples of soil or leaf litter macroinvertebrates were collected using 25 by 25 centimeter quadrats at 4 random locations along each transect line. To collect soil microfauna, for each transect, one sample of soil, about 200 milliliters volume, was collected and placed in a plastic bag for processing through the Tolkien funnel and returned to the lab. To achieve Objective 2, the organisms were sorted and identified up to order for insects using the keys that were provided. The specimens were placed in petri dishes and labeled with their identification and sample type, and the numbers for each organism were counted. To achieve Objective 3, in the lab, soil microfauna sample was placed into the tolerant funnel, and the collecting jar was labeled. The light was left on until the soil dried and the total number of organisms per transect was counted. The data for leaf litter soil were distinguished, and the total number of organisms for each quadrat were counted. The data was assembled per transect. To achieve Objective 4, mean density of organisms and standard error for all the quadrat samples, and the mean density for soil conditions and standard error for all the quadrats were calculated. Graphical representations of the results were also constructed. For the results, soil texture and structure of samples were taken in the two transects. The soil structure was granular and the soil texture was sandy at both transects. Environmental data for samples taken at both transect 1 and transect 2 were graphically represented. Table 4 shows ecological indices for organisms collected from soil samples collected from each transect. Similarly, Table 5 shows ecological indices for organisms collected from leaf litter samples at each transect. Graphs 3 and 4 show the composition of soil arthropods at each transect. Environmental data was collected using random sampling and quadrat sampling based on the transects. There was very little difference between the data collected at the two transects, which indicates homogeneity in the sample area. A variety of organisms were collected, featuring organisms across 20 orders. There was very little overlap between the orders collected from the soil and leaf litter data, as well as between the two transects. This indicates that there is significant variation in arthropod biodiversity. The most abundant order of arthropods found in the leaf litter was Isoptera. where 19 organisms were found, and in the soil samples the most abundant order was Hymenoptera, where 38 organisms were found. This is a stark contrast to data collected by Strickland and his team as well as Yang and Chen, where they found the most abundant order to be Acari. Only two Acari organisms were found in this study. The average density was found to be 6.9 and 3.6 per transect for soil samples, and 4 and 6.5 per transect for leaf litter. The mean density per meter squared was 1.7 and 0.9 per transect for soil samples, and 1 and 1.6 per transect for leaf litter. This low density indicates that while there is high species richness, there is low soil fauna abundance in the forest. In conclusion, the composition of soil fauna in the arena forest was identified. Of the 20 orders of arthropods found, the most abundant individual was Isoptera, and the in the soil samples was Hymenoptera. There needs to be more study on soil faunal composition of the forest in Trinidad, as previously stated by Strickland, such as soil formation, litter decomposition, nutrient cycling, biotic regulation, and for promoting plant growth. Studies should be undertaken at different types of forests across Trinidad, as well as at different ecosystems at different times of the year. Data collected can shed light on productivity of different forests and ecosystems.